So this is going to be a partial key to the gene expression exam review. So question one, kind of our big overview. Generally, there are two main parts to gene expression. First part is transcription, when the DNA is copied into mRNA in the nucleus. And then we have translation, when the mRNA is read to make protein at the ribosome, so out of the nucleus. Try not to get those two mixed up. Make sure you know the difference between transcription and translation. The enzyme that copies the DNA into mRNA is the RNA polymerase. And then for this question, I would look at that diagram, the one that is on the gene regulation practice worksheet. Looks kind of like this-ish, where you have your gene here, your promoter region, your RNA polymerase, and then all of your various transcription factors kind of coming together, general transcription factors for every gene, as well as specific transcription factors just for this gene, all coming together to set up the transcription initiation complex before the copying can take place. And then you also want to consider that in order for any of this to happen, the chromatin needs to be decondensed. So it can't be all tightly wound around its histone, or this isn't going to be able to get to it. It needs to be in kind of one of these bits that is not wound up and spread out so that it can be accessed. And then if any of those conditions are not met, if you don't have all the right transcription factors, if the chromatin isn't accessible, the DNA will not be able to be copied. Number 5, post-transcriptional modification. So between transcription and translation, the mRNA gets modified, and it gets modified by splicing, which is cutting out the introns and leaving the exons, gluing those back together. We have the poly A tail on the 3' end and the modified guanine cap on the 5' end. And then with alternate splicing, which plays into this part, there are some genes that can be spliced in different ways. So different parts can be removed or left in out of the same DNA sequence, and that is going to end up resulting in different proteins or sort of different versions of a similar protein. 7, we're getting to the non-coding RNAs. So remember that not every bit of DNA that is transcribed is actually going to code for a protein. Some of that is going to be RNA that's used in gene regulation. One of the ways that it can be used is it can actually bind to the DNA itself and sort of block or prevent transcription of that gene. Another way is that it can bind to the mRNA transcripts that have been produced, which will either just stop it from being translated, or it will attract an enzyme that will destroy that mRNA transcript. So RNA can interfere with the transcription or translation of the gene at various stages in the process. Okay, so now let's look at the translation process. We have our mRNA transcript, and then we have the ribosome, which is going to kind of clamp on to the mRNA transcript in two pieces. So we have the large subunit on the top and the small subunit on the bottom. And then this ribosome has 3 sites for tRNA: the A site, the P site, and the E site. So the ribosome is going to kind of scan along the mRNA until it finds our start codon, which is AUG. So if we look more closely at a tRNA, which is also made of RNA, we have this structure with the anticodon, which is 3 bases at the bottom. Let's pretend they're A, I don't know, they could be any 3 bases. And then at the top, it has the corresponding amino acid. Realizing that's a lot of A's now, but whatever, we'll go with it. So these tRNAs go into the slots on the ribosome, starting with the A, and then as the ribosome progresses, they move to the P site and then the E site. So if we look at a translation in progress, we're going to have one of these at the A site with its amino acid, one of these at the P site holding an amino acid as well, and this one is going to have the growing chain of amino acids on it, and then one at the E site, which is already bonded its amino acid onto that chain, like this one was its amino acid, and it is getting ready to exit. And then as this is happening, a bond is being formed between the amino acid at the A site and the amino acid at the P site. So when these all slide on over, this one leaves, this one goes to the E site and detaches from these, and then they all end up attached to that one. And then here, if we look at mutations, so mutations are just changes in the DNA, and point mutation means a change of a single base. So we can have an insertion, one base gets put in, a deletion, one base gets taken out, or a substitution, one base gets swapped for a different base. We would expect insertions and deletions to have the largest effect because remember the bases are read in sequence of 3. So if I just make up something A, G, 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 C, 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 if these were my sequences of 3, but I put in, say, an extra A right there, suddenly my sequence is A, G, 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 C, C, and see these are going to be completely different amino acids than these are, and so we're going to get a whole different protein than what was intended, and it probably will have the wrong shape and then won't function. Substitution could have a large effect, but then again it might not. Oh, and we refer to these kind of mutations as a frame shift, when the reading frame of the codons shifts over. We can also have mutations that have no effect. We can have silent mutations because some of the codons are redundant. There are some amino acids that have more than one codon. There's some that have like 4 different codons. And so sometimes it's possible to change a base, particularly if it's the last base in the set of 3, and still end up with the same amino acids. So again, that's a silent mutation. Next, transcription. So transcription is when we take the DNA and we create the complementary mRNA to that DNA. And so you have to remember that C pairs with G, A pairs with T, but then in RNA there are no T's, there is U instead. So if I'm going to do this whole sequence, so we do A, G, U, U, A, U, G, C, A, C, C, U, G, 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 U, G, running out of room here, C, U, C, A, U, A, G, C, C. Right, the next step is going to be translate. So we're going to take this using the codon chart, which you will get for the exam, and translate it. But I need to remember that the first codon always has to be AUG. So I'm going to start scanning along this until I find, here's my first AUG. So that's my first set of 3. And then here's 3, here's 3, 3, 3, 3, 3, and we've got a C left over. Right, so I pulled out a codon chart to translate this, And then UAG right here is a stop codon, which means the translation is going to stop at that point. The ribosome is going to disengage from the mRNA, and then this new amino acid chain, a polypeptide now, can go to be folded into its correct protein shape. Okay. General structure of an operon. Somewhere we've got a regulatory gene, then maybe we have some more DNA, and then we have our actual operon part, where we have the promoter, the operator, and then the gene. And so potentially a repressor protein can be bound to the operator, and if the repressor protein is bound to the operator, then the RNA polymerase is not able to transcribe. If the repressor is not bound to the operator, then the RNA polymerase is able to transcribe. We have two different options with the lac operon. If lactose is there, lactose binds into the repressor, and the repressor changes shape and falls off, the gene is transcribed. If lactose is not there, then the repressor is bound to the operator, get rid of lactose, and the gene is not transcribed. This gene makes enzymes that digest lactose, so it makes sense if there is lactose there, we would want those genes to be turned on so that we can make those enzymes to digest the lactose. But if there's no lactose, there's no reason to be making those enzymes, so that gene should be turned off. The trip operon is also an operon, so it has the same basic structure, regulatory gene, some other stuff. Promoter region, operon, genes, and in this case, this is a gene that makes tryptophan. So, when tryptophan is there, tryptophan binds in the repressor protein, causing it to be able to bind to the operator, blocking gene transcription. When tryptophan is not there, then the repressor comes off, it doesn't fit, and the gene is transcribed. So it's kind of the opposite of the lac operon gene. And this again makes sense because you don't want to be making a bunch of tryptophan if you already have tryptophan. So tryptophan is able to shut down the gene. It's a negative feedback process. If you don't have tryptophan, then you might need to make tryptophan. So it makes sense that the gene would be turned on in that scenario. Oh, and the function of the regulatory gene is to make this repressor protein. Transformation in bacteria is what we did in the lab. It's when a bacteria can take in some genes from outside the environment and use them for itself. Methylation is adding methyl groups to the histone proteins of the chromatin, and what those do is cause the chromatin to coil up tightly and condense, so the RNA polymerase is not able to transcribe those genes, essentially turning those genes off. And we kind of refer to those as tags. Sections of the genes that are uncoiled are able to be transcribed. So this sort of remodeling of the chromatin is how cells are differentiated. Certain genes called master regulatory genes can be turned on or off depending on what type of cell that cell is going to become. So we have stem cells, which are cells that could become any kind of cell, and those are cells that are present in like very early developing embryos, but eventually these tags are going to get attached to the chromatin, so the cells are going to get differentiated. They'll be designated like you're going to be part of the brain, or you're going to be part of the digestive system, and the cells start getting more and more differentiated as that embryo develops. You do still have some stem cells in you. For example, you have stem cells in your bone marrow that are stem cells for different types of blood cells. So they're already modified that they can only become blood cells, but they could become red blood cells or white blood cells or platelets, depending on how that cell is modified after it's produced. Restriction enzymes are enzymes that chop DNA into little bits called fragments, and they always chop at the same sequence. So each restriction enzyme has a specific sequence that it's going to cut the DNA at. This is useful for biotechnology because we can use it to identify DNA and see if it matches another DNA sample that we have, because the same person's DNA will always be cut into the same pattern of fragments, whereas a different person's DNA will be cut into a different pattern of fragments. Gel electrophoresis involves taking those fragments, inserting them into a gel, running a current through the gel so the DNA fragments migrate down the gel, with the larger fragments moving slower than the smaller fragments because it's just harder for them to get through that thick gel. So this allows the fragments to spread out and will create a distinct pattern depending on whose DNA was used. And finally down here, PCR is the process of amplifying a DNA sample by changing the temperature and putting in certain conditions that induce it to replicate itself. So you can exponentially increase a DNA sample so that you have enough to work with. Right, so this is just a quick overview. If there's parts that you're still confused on, I recommend you watch the videos from them again and or ask me questions over Schoology or in class.